share your presentation and let us start.
I think they have done something with the WebEx. It is looking totally different today. Are you able to see my presentation? Can you see my presentation? Hello? Can you see my presentation? Oh, so but can you see my slides? Okay, then I'll start. Okay, okay. Good afternoon, everyone. Sorry for this delay. This technology, sometimes gives you pain instead of helping you. So never mind. So today we are going to cover NMR metabolomics. In last lecture we covered the in vivo spectroscopy, wherein we looked at how different NMR sensitive nuclei can be used to give information about the living systems. Today we are dealing with a subject which is called NMR metabolomics. It is a form of in vitro spectroscopy, and we'll see how in vitro spectroscopy is useful in developing biomarkers of disease, and also looking at the therapeutic response of different interventions. So if you look at the pyramid of life, we all know we have about 25,000 genes, and there's we are also talking about proteins. We have proteins, then we have metabolites, and and every these all type of studies are done using omic techniques, and we have genomics, transcriptomics, and the proteomics, and then finally the latest in the omic machinery is metabolomics, which gives you information of the phenotypes of the individuals, and gives you information about the metabolic profiles and disease processes which are happening in vitro. And then you can draw any sample from the humans, the tissue or the urine or serum or saliva or CSF, whatever you is available to you. You can subject them to NMR spectroscopy and do the metabolomic studies. And if you look at the metabolomes of life, all mammals have about 60,000 biochemicals. And when you go to all microbes, there are about 100,000 chemicals. And most complex is the plant family, where we have about 300,000 chemicals which are available for metabolomic research. So, and metabolomic is the highest platform which gives you information about the these three topics along with bioinformatics. Can you give a good approach to system biology, and you understand the disease process and the metabolic processes which are happening in the body in health and disease. So, like now let us think what a metabolite means. Metabolites are the end products of cellular metabolic and regulatory processes, and their levels can be regarded as the ultimate response of biological system to genetic and environmental changes. And any organic molecule detectable in the body with a molecular weight of less than 1000 Dalton, we categorize them in the category of metabolites. They include peptides, oligonucleotides, sugars, nucleosides, organic acids, ketones, aldehydes, amines. I think this is a huge, category of chemicals which one can which categorize under metabolites, and this also includes human and microbial products. And usually the concentration of these metabolites is higher than 1 micromolar, and so they are at a very lower concentrations. You need to have a very high sensitivity technique to detect these metabolites in the living tissues. So metabolomics is coined as by Nicholson and Linden. They coined this term metabolomics. Like I said, it's another omic technique, the latest omic technique, and is a field of life science research that uses high throughput technologies to identify and or to characterize all these small molecules or metabolites in a cell, tissue, or a whole organism in health and disease. And it also gives you the simultaneous and rapid systematic determination of metabolic levels in body fluids, so you can use the whole organisms or any body fluid or the cells, or and then maybe the the tissue extracts, and then see when you expose these biological tissues to an external stimuli, how does the metabolism and the metabolic profile change. So this is a very sensitive technique, and here I am showing a urine spectrum, untreated urine spectrum for humans at 900 megahertz. You can see the number of peaks. You can see there's a huge number of peaks which are visualized over here. And if you look at this urine spectrum has a lot of information contained in it. It has biomarker information on genotypes, the physiological state, the nutritional state of this person whose urine is this, the gut microbes, the biological age, and presence of disease. And it also has translatable biomarkers like diagnostic, prognostic, toxicity, and efficacy. So there's so many information which is available in this urine spectrum. And based on the this is a study where people took hundreds of urine samples from unknown population, and they could categorize these populations based on the urine samples by doing a NMR metabolomics, and they could separate out these two populations just on the basis of the metabolic profile of the urine. So these were population from American, Japanese, and Chinese, and they found how beautifully these population can be separated based on the urine profile. The change in the metabolic profile because of the genetic origin, the food habits, and environmental factors. So that's how it's so sensitive that just on basis of urine you can separate out these populations. And metabolomics had advantages that it can measure multiple of metabolites at once, and you can go for tens to hundreds of chemicals you can measure at once, and you can allow metabolic profiles or fingerprints to be generated. And most of the metabolic approaches are automated, and they are relatively little simple, and you can the sample preparation is not so difficult or so complex, and it can be also quantitative if you are using NMR metabolomics, and analysis and results can be in less than 5 minutes. So these are some of the metabolomic techniques. Most of the people who are chemists, they know UPLC, HPLC, the microfluid C, the LCMS, FTMS, QTOF, MS, and NMR spectroscopy, X-ray, crystallography, and GCMS. There are several techniques which can be used to study metabolomics. But when you look at the sensitivity patterns, of course, LCMS has the highest sensitivity, where it can go up to micromolar concentrations. NMR that way is doesn't have so much of sensitivity. It can go into millimolar to micromolar concentrations, but definitely LCMS is a gold standard for doing the metabolomic studies. However, NMR has certain advantages which you will see in due course of time. So if you look at the comparison between LCMS and NMR, the detection limits, as I said, uh, it is not as good as MS, where you can have a picomolar concentration one can pick up through mass spectroscopy, whereas in NMR, you can go up to millimolar or micromolar concentration. Of course, you can go to nanomolar if you use cryogenic probes, which have very high sensitivity of detection. And then universality of metabolite detection. If in NMR, if metabolite has hydrogen or NMR sensitive nuclei, then you can detect these nuclei, and sometimes because of the protein and micromolecules, there could be the line broadening, which can affect your results. However, MS is that way quite better, but in MS you need to first separate all components and then quantify them. So it's a little tricky. Sample handling is you can use the whole sample, analyze it in one measurement. That you don't have to work too much on the sample preparation, and sample quantity is typically 200 to 400 microliters, but less for microcoil probes, up to 5 to 10 microliters. But however, in MS you need very low microliter range sample volumes. Sample recovery is in NMR, it is non-destructive. That means you can use this sample for NMR and then store it back into cryogenic temperatures, and you can subsequently use it many times in future. How in MS the sample is destructive, and you cannot use it again because you are already consuming the sample. Analytical reproducibility is very high in NMR, and it is fair in in MS. Sample preparation is minimal. Addition of buffer, D2 and chemical shift reagents, and can be in MS. The sample preparation can be substantial. Often needs different LZ columns and protein precipitation, chemicals and methods. And ease of molecular identification high both in database automatic material, and you have databases where you can easily refer to the database and find out what are the molecules present in the sample. And time to collect for basic data is 5 minutes for NMR. It is minimum 10 minutes for UPLs or MS thing. The quantitation is 1 to 5%, and robustness of instrumentation is very high in NMR. NMR instruments are very robust, and there are hardly any breakdowns, in the NMR techniques. Whereas in robustness of instrument is low in MS because of the maintenance of the columns and other sub parts of the equipment. Then molecular dynamic studies is possible by T1, T2 reaction times and diffusion coefficient measurements, which is not possible using mass spectroscopy. Analysis of tissue samples is possible by using magic angle spinning NMR, which is not possible by MS. So there are several advantages when you do NMR metabolomics compared to MS, and most of the things it is much cheaper than the MS technique. So there are two routes to metabolomics. One is quantitative methods, one is chemometric patterns method, wherein you can do several samples, and then you can, by chemometric methods, you can categorize these samples according to some special characteristics, whether it's a disease or healthy. You can, or you, if they are exposed to different environmental conditions, or if they are from different genetic origins, you can separate them through chemometric methods. And quantitative methods, you have a spectra and then you can quantify each metabolite individually. But in chemometric methods, you find out some principal components of metabolites which can affect into the categorization of these samples in different clusters. So these are the two methods of metabolomics. So, why is metabolic relevant? Because it generates metabolic signatures. It monitor measure metabolic fluxes in the metabolic pathways. They can also monitor enzyme pathway kinetics, and they can identify and assess phenotypes and monitor gene environmental interactions, track effect from toxin, drugs, and surgery. You can expose a tissue or living system through toxin, drugs, and surgery, and see what are the effects of these toxin and interventions on the tissue.
Sir,
Yes, sir.
this slides are not moving, looks like.
Really?
Yeah, sir. Yeah. Represent the screen.
Till which slide you saw?
The first slide only is still.
Oh, but why so?
Now it is moved. This
Now
fourth
you are able to see this slide?
is seventh slide, no? Ah, yes.
This is
So
seventh slide.
yeah, so in between
Did,
it didn't move.
did you saw this before?
No, sir. First slide and then suddenly this slide came.
Do you see this slide?
Yeah, this slide is in.
So shall I go back?
You can just brief it. What you
Okay, I
cover?
said this slide you saw.
This is seventh slide. Yeah, we can see that.
No, this slide. Did you see this slide? First slide.
No, it's not coming again. It's becoming slow.
But this no, you can't see slides now.
This is still in which slide? Stuck at seventh slide. Now we can see the seventh slide, sir.
No,
Now we didn't see the changes.
you are seeing changes.
No.
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 So only seven slide is visible.
Only seventh slide is visible. So you can represent the screen.
Can I reconnect?
Ah, yeah, you can reconnect.
Yeah. Okay.
Screen. That issue. Hmm? Reconnect or PPT, reshare the PPT. I think that should work. Slow or what? न्यू इंटरनेट इंस्टॉल किया है अभी तो है. Yes, sir. We can see it now. You just move 1 2 slides. Let's see whether it's moving.
Is it moving now?
It is not. Click 2 3 slides and then come back again. Maybe. Yeah, it's moving.
Are you able to see?
Yeah, now it is moving.
Okay.
Okay, thank you, sir.
So I was just talking about this pyramid of life. You have, genomes, transcriptomes, proteomes, and then latest is highest metabolic profile is the metabolomes. Where you have several biochemicals which define the phenotype of the of the living system. So we have about 25,000 genes. Then over that you have 2,500 enzymes, and 1,400 chemicals when you define a human metabolome. You see how these omic techniques can define the phenotype of a living system. And then we in if you look at the different metabolomes of life, you have 60,000 chemicals when you talk about mammals, and when you talk of microbes, you have 100,000 chemicals. But plants are very complex, and you can see 300 chemicals. You can look at these number of chemicals through metabolic approach when you are dealing with plants. So plants are very complex metabolome, and one can really study them using NMR based metabolomics. So this, metabolomics, proteomics, and genomics together with bioinformatics and chemoinformatics can enable the study of system biology and understand the living system in health and disease. So metabolites. So what are the metabolites? Metabolites are the end products of cellular metabolic and regulatory processes, and their levels can be regarded as the ultimate response our biological system to genetic and environmental changes. And any organic molecules detectable in the body with a molecular weight of less than 1,000 Dalton, we categorize them in metabolites, and which includes peptides, oligonucleotides, sugars, organic acids, ketones, aldehydes, amines, so many variety of chemicals which are there present in our body, and which can be categorized under the category of metabolites. And they also include human and microbial products, and generally concentration is higher than 1 micromolar, though there are some metabolites which are at lower concentration as well. But generally, this is the concentration most of the these metabolites are present in. So the metabolomics term was, coined by Nicholson and Linden, and this is another technique like omic techniques like genomics and proteomics. It's a field of life science research that uses high throughput technologies to identify or to characterize all the small molecules or metabolites in a cell, tissue, or whole organism in health and disease. And this also can also do a systematic determination of metabolic levels in body fluids, whether it is a serum, blood, urine, CSF, or any other body fluid, and or the whole organism or cells or tissues, over time, and as a consequence of any exposure to this living system by an external stimuli, one can study using metabolomics. So this is a untreated human urine spectra taken at a 900 megahertz, NMR spectrometer, you can see the number of chemicals you are seeing as a peaks, and this spectra contains lot of information about the genotype, the physiological state of the person, the nutritional state of the person, then the gut microbes, biological age and presence of disease. This spectra can give you all these information about these biomarker information, and then you can also have translatable biomarkers on diagnostic, prognostic, toxicity and efficacy. And you will be surprised. There's a study where just on the basis of urine they have taken randomly urine sample for hundreds of volunteers, and when they subjected that to NMR spectroscopy and doing chemometric methods, they could separate out these populations, Japanese, American, Chinese, just on the basis of their urine sample. So this you can see how sensitive this technique is, that it can detect all your latent information about your genetic origin, or your nutritional state, your environmental factors, all these things, your food habits, and it will categorize you in different clusters based on the metabolic profile. So this is a very sensitive technique to detect minor, even minute changes which are happening in metabolic processes in the body. So the advantage of metabolomics is you can measure multiple metabolites, 10 to hundreds of metabolites at once, and you don't need any separation of these chemicals like you do in some other techniques. And it allows metabolic profile or fingerprints of health and disease. And mostly techniques are automated, and they need relatively very less sample preparation. And these can be quantitative. You can actually quantify each metabolite or the biochemical present in the body, and analysis and results can be as fast as less than 5 minutes. And let me tell you, they can be high throughput methods to do disease detection when you have mass casualties. This can be a method which can do a high throughput diagnosis of the casualty patients. And these are the different techniques which are used for metabolomics: HPLC, UPLC, the LCMS, FTMS, QTOF, MS, NMR spectroscopy, X-ray crystallography, GCMS, FTIR. These are all techniques which are present, available to you to do metabolomic techniques. But most of them are, uh, the main comparison when you do metabolomics is between the MS, LCMS, and the NMR spectroscopy. These are the two techniques which are being used very commonly for this metabolomic work research. And if you look at the sensitivity of these different techniques, no doubt the LCMS has the highest sensitivity, and it can pick from nanomolar to sometimes picomolar concentrations. This is a very sensitive technique, and I would say this is a gold standard for doing metabolomics. But when you look at the NMR, though you can't see concentrations, you can see only concentration between millimolar and micromolar concentrations, but still it has several advantages which make it again a modality of choice for doing metabolic studies. So this is some of the comparisons. You will come about them in future when we have further slides. But most of the important thing is detection limits. Definitely MS is much better than NMR as far as detection limits are concerned, because you can measure up to picomolar concentrations, whereas in NMR you can go from micromolar to maybe sometimes if you have a cryoprobes you can go to picomolar concentration, but very rarely you can do this. And sample handling is much easier in NMR compared to MS. Then sample recovery, you can this NMR technique is non-destructive. If you use the sample today, you can preserve it and use it after 3 months or 6 months. But in MS, the sample is restricted when you separate and then use it for the analysis. And then reproducibility is very high when you compare to NMR with LCMS. And then ease of molecular identification. NMR has a full database of biochemicals which can be used, and you don't need to know what are the metabolites present. Whereas in MS, you have to look at the columns and other chemicals to be selected depending on the type of study, whether you're doing a polar or non-polar or whatever metabolites you are studying, you need to have some prior information. Time to collect basic data is 5 minutes for 1D NMR. It is 10 to 15 minutes for UPLCMS. And quantification can be done in most of the cases in robustness of instruments. NMR spectrometers are very robust, and generally they don't break down so easily, and it is very high robustness is there, and you can study molecular dynamic information using NMR based on T1, T2 resident times and diffusion coefficient measurements, which is not possible using MS. And then analysis of tissue samples you can do using mass. Mass is magic angle spinning NMR, which is not possible using NMR. So there are databases available in MS. Complete database is available. However, MR also has a database available, but still many more additions are required to this database based on NMR detection of some of the molecules at lower concentration. So it has two routes of metabolomics. One is quantitative method, where you see spectrum like this and you can quantify each peak separately. The other is chemometric pattern method, wherein you have two different populations of several populations which have different characteristics. So you can divide them, you can categorize them in different clusters depending on the whether they are disease or healthy, or they are being exposed to different stressors. So you can actually see if they are exposed to suppose heat stress and for a longer time, then you can see two different clusters and along compared with the healthy volunteers. So this is basically a pattern method where the these samples can be classified as per the changes in the metabolic levels due to external factors. So metabolics is relevant because they can generate metabolic signatures, they can measure metabolic flux, and they can do the pathway kinetics, and they can identify phenotypes and monitor gene and environment interactions, and then also track effects from toxin, drugs, and surgery. And if you have any intervention, you can track how this has the intervention has affected the these samples of the living systems. So there are several applications of metabolomics. It is used in toxicology testing, clinical trial testing, fermentation monitoring, food and beverage test, nutraceutical research, drug phenotyping, water quality testing, organ transplantation, stress response monitoring, genetic disease test, nutrition analysis, clinical blood analysis, clinical urine analysis, cholesterol testing, drug compliance, dialysis monitoring, petrochemical analysis. So it has several large number of applications. These are only, this is only the tip of the iceberg, but you can imagine any number of applications using this metabolomic approach. So in medical field, it is very important because it can generate metabolic signatures or disease signature of a particular disease, and you obtain a more holistic view of metabolism, and then look at the treatment modalities, and it can accelerate assessment and diagnosis. As I said, they are quite fast, and they can be rapidly and accurately measured, and you can make a high throughput devices in a situation where you measure mass casualties or mass exposure to any hazardous chemicals or gases. You can monitor the status of different people depending on the their severity of their exposure by using high throughput metabolomics, and then you can rapidly track effects from drug and surgery. So there's a human metabolome project of about 7.5 million project where in Canada it was taken, and his mandate is to quantify normal and abnormal ranges of all metabolites in urine, CSF, plasma, and void blood cells, and this to make this data available to everybody free of cost and electronically accessible to the globe. And this is purpose is to facilitate medical metabolomics, and objective is to improve disease identification, disease prognosis and prediction, disease monitoring, drug metabolism and toxicology, linkage between metabolome and genome, development of software for metabolomics. This is all the mandate of this, the objective of this human metabolome project. And so many places now people are detecting a single metabolite based biomarker of a disease. Like if you look at the marker for sugar, the diabetes, sugar is the one which you will always find, which can distinguish two persons based on the glucose levels in urine or sugar. Similarly, for other disease condition also, you can have one or couple of metabolites, a group of metabolites which can define that disease and give you a fingerprinting of that particular disease. So these are large list of components, chemicals which can be seen using NMR metabolomics, and you can also create a metabolic microarray. In this is in the cancers. You can have patient and versus the metabolites, and you can look at this array and look at which metabolites is increasing, normal, below normal, above normal, or ascent in the different patients. So you can actually generate a metabolome microarray like this, and look at the a group of patients, a cluster of patients in one single go. So NMR based metabolomics, you can have biological tissue samples, then you extract it, and then you do the tissue extracts or the body fluids, and then do a chemical analysis, and then you have different you get peaks, and then you can label these peaks depending on their chemical shifts. You all know that every chemical has a very distinct chemical shift. Depending on that, you can label all the peaks which you see, which you look at during the in the spectra. So when you talk about NMR metabolomics, body fluids, you can use urine, blood serum, CSF, seminal fluids, bile, synovial fluids, which are in the joints, and then you can also do cell cultures and then tissue extracts. All these samples can be used to carry out high resolution NMR spectroscopy or the metabolomics. So the metabolomics literature is growing very rapidly. You can look at in 1999, you have hardly any publications in on metabolomics. You can see how exponentially these number of publications have increased over these years, and they are still increased because of its popularity and the importance of this technique in disease diagnosis. So the NMR-based metabolomics has advantages over other LCMS and other. It's a very stable and robust instrumentation. It is non-destructive, quantitative, and minimal sample preparation. No separation of components needed as you do in LCMS. The results are very highly reliable and reproducible, and it is much faster and easy to use. It can be used as a very high throughput device technique. Detect sugars and non-ionizing compounds, and it can give you facile measurement of flux. Allows work with living cells systems. You can measure, do the real-time kinetics of the reactions. Gold standard for unknown IDs. If you do not know what type of metabolites or the chemicals will be present in a sample, it is a gold standard for that. It will give you a complete profile of the sample, and it has much cheaper operational cost than MS, and they are easily available because NMR spectrometers are available in almost all the chemistry departments of any institute or the university. So these type of studies are much easier to carry out. So we know again this is again a reiteration of the fact that if you want to have better sensitivity, you need to have higher field strength, magnetic field, and most of the places you have 400, 500, 600, and 800 megahertz NMR spectrometers which are available, and now even 1 megahertz, 1000 megahertz, 1 gigahertz NMR spectrometer is also available, and the cost can vary from 1.5 to 16 crores for this type of equipment. And for metabolic profiling of these bio fluids, you need to do one dimensional proton spectroscopy to look at the profiles. And if you look at the human biofluid records through NMR, you have identified 67 individual components have been identified, and all the 67 have been quantified also. In LCMS 245, you can see how large number of LCMS has detected because of the high sensitivity of LCMS over NMR. Then in CSF, you identified 53, biochemicals, and all the 53 have been quantified. Similar way in LCMS also, you can see there's a comparison only between NMR and LCMS. You can see 78, and then 78 have been quantified. When you look at the urine, out of 209 which have been identified, all the 209 have been quantified in the NMR spectroscopy, whereas in LCMS, 219 have been identified and only 93 are quantified. You can see how NMR scores over LCMS when it amounts to quantification of the biochemicals. And these are the metabolites which can be observed by NMR: the amino acids, organic acids and bases, nucleotides, carbohydrates, osmolytes, lipids. All these can be very well visualized using NMR. And these are the some of the in serum. If you have a blood sample, you can look at all these amino acids: lysine, glutamate, glutamine, aspartate, alanine, leucine, valine. All these metabolites can be seen, and ketone by acetate, LDL, VLDL, membrane beta-choline, phosphocholine, for ethanolamine. All these large number of metabolites can be visualized when you are looking at the serum samples. Similarly, when you look at the and these are some of the properties of these biochemical serum creatinine. There's a late stage organ stress and tissue breakdown. When serum creatinine increases in blood, there's a distress in the kidney, and when there's a chronic renal failure, serum creatinine goes very high. So similarly, all these metabolites have certain properties, and they depict a particular type of disease. But like if you have citrate, lactate, and acetate acetone, it shows oxidative stress, apoptosis, anoxia, ischemia. So based on these metabolic concentrations, one can detect the type of disease a person can have. So experimental methods are, metabolic levels, as I said earlier, are easily affected by diet, age, BMI, gender, and time of the day. So you should account for these, parameters when you take the samples from the humans. And it is always preferred to take a 12 hours fasting when you take the sample, because the dietary, whatever you have taken last night can affect your sample. So it is always to take sample, during fasting. Then biofluids and tissues are metabolically active. So you should, immediately as soon as you draw the sample, after some time, you should, cool them to a freezing liquid nitrogen temperature so that these samples don't degrade. And you should immediately, as soon as you draw the samples, should label them so that you don't mix the samples together. And you must take some more time to do the experimental design. Then otherwise you will waste a lot of fruitful time in data processing, and then finally you find there's some error in the experimental design. So it is always good to give some time to some good thought to an experimental design to have a good experiments. And these are some of the applications which I want to show you regarding the studies which we have done in where you can use them as a the high throughput devices. So this is the complete workflow of the metabolomics. You can look at the energy metabolism TCS cycle, and then biomarker discovery and validation. Then you do the PC analysis and disease classification, and you can do this in animal model, cell lines, or on the whole human samples. The body fluids from humans, and then you can identify and quantify different the different metabolites. So this is if you are using different samples, if you are using a serum or the blood samples, you need a CPMG pulse sequence, and since because serum has a lot of macromolecules and proteins, so you have to use CPMG. And urine is very simple. You can see a normal 90-degree pulse and FID sequence, no ZPR. And then tissue also you need a magic angle spinning and magic angle high resolution NMR, and then you carry out these samples and do the processing to get your results. So this is a spectrometer. I think we have talked about NMR, MR instrumentation a lot. So basically everything is same, except you have a vertical bore, and from the top you can inject the sample. From the here you can inject the sample into the probe and do the analysis. And when it is sample name not by sample process required due to the absence of macromolecules, and hence you can urine sample is very easy because there's no macromolecules and proteins there, so you can directly use them. Only thing you have to do a filtration to filter out any small suspending particles with a 0.22 micrometer filter, and then you can store it at -80 degrees centigrade or in liquid nitrogen temperature. You can add some sodium azide to protect the sample from further degradation, and EDT buffer at 7.4 pH. So because every individual has a pH variation in the urine, it can vary from anything between 5.5 to 7.5 to account, and many of the metabolites they are pH sensitive, so you have to have a common pH value for the sample. So you add a EDTA buffer which has which brings it to 7.4 pH value, and then you add a reference material TSP or DSS, and then carry out the sample at different spectrometers available to you. So I will skip some, and you all know the effect of these parameters on the NMR spectrum. So this is am I shooting time, Professor Pal?
Yes, sir. You can continue some more time to complete it. Or
Okay.
As you feel like.
Okay. So I think this is how you prepare the sample for urine. If you have a urine sample, you mix with 200 microliter of deuterated buffer solution, sodium phosphate pH. This is what I told you about making the pH constant, and then TSP as a internal reference. Then you centrifuge the sample at 2600 G for 10 minutes to remove any precipitates present in the urine, and then TSP is used as a chemical shift reference, and TSP peak will come at 0 ppm, and all other peaks will be measured with respect to TSP. And then water suppression is done using the water suppressed pre-saturation pulses, as we did in vivo spectroscopy, in proton spectroscopy. And then you can subject it to NOSI-PR 1D sequence, and then look at the then you do a post-processing. So I will directly go to the some of the applications. This was in case of any nuclear fallout, there will be a lot of large population which will be affected, and we thought whether we can find any method of doing a high throughput method to find out the radiation dose of exposure to the individuals while there's a nuclear accident or some nuclear fallout. So this was done on the animal model, and this was a male mice, and wherein we had two groups. One was control group, sham irradiated, and then the other group was irradiated by different doses of radiation, like 3, 5, and 8 gray of whole body gamma radiation, and this was given through gamma cell irradiation facility at INMAS. And then we collected blood and urine samples at different time points from these mice animals, and then the samples were stored at -80 degrees centigrade before they were subjected to NMR analysis. So this is a serum spectrum from the serum. You can see group of metabolites which are visualized here, and lower plate you see the spectrum from the urine, and urine gives you a lot of much more metabolites, and it is always good to work with urine because it's completely non-invasive and you don't need a trained manpower, because when there's any accident or any casual eventuality, you don't have medical sub paramedical staff to draw blood. It is always better to work with urine samples so that the subject or the volunteer can, give urine of his own without being, waiting for the biomedical assistance to come. So we always preferred to use urine as a body fluid to do the, this type of research. And then this urine, as I told you earlier, it has complete information about the whole functioning of the different organ, and about the functioning of the human body. It has all, containments there, all the, metabolites containing to different sections of the body. You can see, one can see them, all the functional windows are seen. And then you can see the metabolites related to energy metabolism, protein metabolism, lipid metabolism, gut flora microbiome, membrane metabolism in urine, as well as serum. So all these metabolites are visualized, and then also, in kidney tissues and liver tissues, all these metabolites are very well visualized. So this is where we have shown, this is a, spectrum from the control mice, and then mice which are exposed to 3 gray, 5 gray, and 8 gray radiation. You can see the spectral patterns, and we have seen that there's a radiation induced changes in serum, and increase in lactate, amino acids, LDL, VLDLs, and choline containing compounds, which shows the disturbance in energy, lipid and protein metabolism in these, irradiated rats. And then, you can also look at, we looked at, this biochemical profile or the metabolic profile of these animals at different, irradiation, 3 gray, 5 gray, and 8 gray at different days. And we saw that, day one, there's hardly any, changes in the metabolic profiles. But if you look at the day 3 onwards, we started seeing the, changes in the metabolic profiles. And day 5, they were very prominent in all the three groups. So, and we found significant changes in LDL, VLDL, lactate, alanine, as I said in the last slide. And so that means, the major changes are happening in the tissues after the 3rd day of radiation exposure, which is very well visualized here. All the metabolites have increased. These metabolites have increased on day 3 and day 5. And day 5, they are, fully, the increases very much there. Then, based on these chemometric methods, we were able to, separate these 4, categories, 4 groups into different groups. You can see here. And on day, if you look at day 5, all the 4 groups are very well, identified separately. Otherwise, day 1, there's a lot of only one group a day. Probably the 8 gray group is, separated. The rest all are together in the PCA plot. And gray, day 3, still you have some, separation. But in day 5, you see all the 4 groups. The control group, the 3 gray, 5 gray, and 8 gray, animals are separated out. That means you are able to separate out different population which are exposed to different doses of radiation. And this is a, loading plot where this axis gives you the chemical shift of the particular compound, and this, peak tells you how much change has been happening in this particular metabolite. And these are the metabolites which have caused these are the metabolites which has caused this separation in these groups. So these were the metabolites which were responsible in doing this separation of the different groups of, radiation exposure. So similarly, this is, in urine, and you can see in urine, within 6 hours of the radiation exposure, you are able to detect, changes, and then see the groups distinctly. But when you look at the, after, day 1, you can see all the 4 groups are very well, separated out. So within a day in urine, though blood takes 3 to 5 days to see the reflection of that radiation exposure in urine, within 1 day you are able to see. So within 1 day, if you subject these samples to NMR spectroscopy, you can find out who are the people who have got different doses of radiation immediately. So similarly, these are some of the, then we also did expose the animals only for 5 gray, and then monitored them over longer period of time. We did, pre-exposure 6 hours, 5 days, 10 days, 15 days, 20 days, and 25 days. and took the urine samples, and then these are the spectrums which are shown here at different time points. And these are the metabolic peaks which are seen. And then there are three phases of the radiation exposure: the prodromal phase, the latent phase, and the manifestation illness phase. And you can have a look. This is a same 5 gray in the, this is a serum sample, the blood sample. This is a urine sample, this is a blood sample. And then you can show the these three phases of radiation sickness. You can show the prodromal phase, which is from 48 hours to 6 days. And then the latent phase, where the system tries to, in prodromal phase you have some changes, but then in latent phase the system, the body or the tissues, they tend to recover themselves to some extent. If the exposure is not very severe, they may recover also. But if the exposure is harsh, then they can get into a manifestation phase or the illness phase, where you can start seeing the changes in the body, and then patient or the animal will start degrading, and ultimately may, depending on the exposure, may go to different shutdown processes in the body. And this here it shows the trajectory of these changes from 6. This is a control where no exposure has been given. Then in 6 hours it is going away from the control, and slowly day 1, day 5, day 10. This is the latent phase where the body is, these tissues trying to recover. And then manifestation phase, day 20 and 25, and it is again the metabolic profiles are very different from the controls. And then this, you can say, they've gone into a manifestation phase or the illness phase. Similar thing is in serum. You can see day 25 they're completely into sickness phase, and also in urine. So otherwise here also you see day 10 they are in the pre-dose condition. That means they are in the latent phase of the radiation sickness. And then you can also look at what are the metabolites which have shown changes. And then based on these changes, we found there are 4 metabolites: the succinate, the creatine, the TMO, and the taurine. These are the 4 metabolites which showed us dose-related changes in these animals. So, and then that means, and most of the times we found the reliability of measurement of taurine, which the R is 94% accuracy is there. And of course others are also high, but creatine and taurine were the two biochemicals which had shown high correlation with the doses. And then one can detect, one can make a taurine sensor, where on which if you drop a uh, drop of urine, you will know whether the person is exposed to radiation or not, or to what extent the person has got exposed to a radiation exposure. So based on all the four metabolites, all these categorization in the these groups were done because of the major changes in these 4 metabolites. Then you can also study the thermal stress, heat stress, and the cold stress. And this is a climatic chamber wherein you can create temperatures up to minus 40 degrees centigrade, and up to 55-60 degree positive temperatures Celsius. And then also you can create high altitude for as good as whether even if you are like you are in Leh or Ladakh, you can create high altitude conditions within this chamber. And then you can expose animals in different levels of high altitude or the cold stress or the heat stress, and study their metabolic profiles. And here this is a the control animal, and this animal who has been exposed to cold stress. You can see many of the energy metabolite uh, uh, biochemicals, or the metabolites have had disturbances, alteration in their concentrations when they were exposed to cold stress for minus 10 degree for 3 hours every day. And similarly is when you give cold chronic stress, you can see that these animals, these are the control animals. And this animal on day 1, they are able to separate themselves from the control. And when you measure for day 5, you take sample on day 5 and day 10, day 15, you can see all these times points, they are separated from the control control group. So this shows separation because of the because of the cold stress on these prolonged cold stress on these animals. And now we also wanted to see what all can work to avoid the ill effects of cold stress. So we did a herbal interventions with Tinospora cordifolia, which is a adaptogenic herb. And when we gave this adaptogenic herb to the animals, and these are the control group and group which was not giving any adaptogenic herb, you can see after the cold exposure, these group are very separate. The control group and the cold exposure group are very distinctly shown here. But in the group when the Tinospora was given, you can see you can't distinguish between the control group and the cold stress group. That means the cold in the Tinospora cordifolia has a adaptogenic uh, you know uh, effect, and it can avoid the ill effects of cold stress on on animals. So similar trials can be tried on the humans, and maybe the people who live in high altitude areas, particularly the soldiers and jawans who are in Leh Ladakh or Siachen, they also you don't realize they also get deficit because of low oxygen concentration and these other stressors. Their cognitive parameters get affected. So by giving these interventions, these adaptogenic herbs, one can avoid the ill effects of courses on these jawans. Similarly, you can see also these heat stress where the two animal groups can be separated, and you can actually measure what is the extent of changes, and then you can plot correlation curve between the heat stress and the metabolic concentration, and develop some sensors to detect these changes happening on these. This is 45 degrees centigrade for 3 hours was rats were given to this acute heat stress. So then this is nowadays we talk a lot about gut health, because now people start talking gut and gut brain axis, where now people feel gut health is most important for our health, mental and physical well-being. So this has been used. NMR spectroscopy has been used for looking at the gut health in a colorectal cancer. They have diagnosed colorectal cancer cases based on the metabolomic approach by looking at different stages of colorectal cancer, the stage 3, stage 4, stage 2, and this is the healthy control. And they have been able to quantify these uh, parameters in different stages of colorectal cancer. So similarly, is urinary tract infection. You can see the two groups, and two groups, the healthy group and the people who had urinary tract infection. They can be easily separated. And these are the two metabolites which become the biomarker for urinary tract infection. You have TMA and the SH8. So yeah, and they were identified as optimal candidates for biomarkers for UTI diagnosis. And you can show here the changes in E. coli and non-E. coli parameters with the control group. And very nice technique. A lot of people also tried in metabolomics in the liver transplant, and they have been able to save many lives, because some of the transplant patients, they were dying after transplantation of few weeks of transplantation, because the increase in ammonia in the body, and they detected this ammonia through NMR, and wherever there was a increase in ammonia levels, they treated the patients, and the patients could be saved from fatality. And now my concluding remarks are, metabolomics is rapidly becoming the new clinical chemistry. Metabolomics complements genomics, proteomics, and histology. Metabolomics allows probing of rapid physiological changes or events that are not as easily detected by microarrays or histological methods. It is emerging as an automatic high throughput method for fingerprints of stressors and disease conditions. As I said, these in case of any nuclear accident or any other casualty where large number of people are affected, so it is very easy to draw urine samples from large population and subject them to NMR spectrometry, and and large number of spectrometers, and you can do maybe 10,000 samples within a night. Otherwise, the other methods which are available for doing radiation sickness measurement, they are operator based, and they take at least 46 hours for one sample to be to be to diagnose. So here's a method which is high throughput, easy to use, and you don't need, they're fairly automated. You don't need an operator for to look at the slides, histopathological slides on anything else. So it is a very fast upcoming technique in medical diagnosis and medical research. I think thank you so much for patient hearing.
Yeah, thank you, sir. So participants, any questions? You see the YouTube also? YouTube. No question. Yes, sir. I think as such, no question. Was a question?
Yes, please.
Yeah, I think participants not asking anything.
Okay.
So thanks for the sending this reading material. So we have already shared it with the participants. Let them go through it.
I am sorry for the delay today, but anyway.
It doesn't matter. It happens sometimes. It's not anyone's self. I don't know. It happens sometimes. And so you would like to send some more reading materials, or this will be sufficient for whatever you send?
I may do that.
You may send some new.
I will now send some articles for reading.
Yeah, please send whenever you feel.
Some basic articles.
Whenever you feel time, send, let them